gap in KBase. And there are so many presenters, so I'll go real quick. Just to give a very high level idea, is that the reason for having this app is that when we construct metabolic models, it's a metabolic network. And usually when we construct a metabolic network, there are gaps in that metabolic network due to knowledge gaps. Some genomes are not getting annotated, and so it doesn't, those annotation does not propagate to biochemistry, so there are metabolic gaps. So usually the gap filling algorithms try to fill those gaps in different ways, and many times it uses the minimal set of reactions to fill those gaps. And those minimal set of reactions, most of the time it's biologically irrelevant, or it's far from realistic biology. And so it could be two reactions that fill these gaps, but sometimes realistically it could be 5 reactions that actually represent in actual organisms. So the OMEGGA app is actually trying to do this gap filling more intelligent pattern, that it uses the phenotypic data, the multi-omics data to fill these gaps. And Hyang, later in this presentation, will go into nut and bolts in the OMEGGA apps. And in order to get these intelligent gap filling reactions and associate these reactions with annotations, you need to annotate a genome with multiple annotation algorithms. So the whole pipeline is in a KBase narrative. And Alicia, you can share the link to the narrative. So it looks like this. So anybody can, I'm going to show this real quick. Anybody can create an KBase account. It's really quick, takes 30 seconds. If you go to kbase.us, you can sign up for a KBase account. It takes 30 seconds. And then when you sign up for an account, it comes to this KBase dashboard. And here you can see your narratives that's shared by some other users, that public tutorials. So this workflow that we are presenting today also appears in the public tab. And you can also access these workflows because Alicia is putting those links in the chat box. And we also have the HTML version of this narrative. So to access the HTML version of the narrative, you don't need a KBase login. You can access the narrative right away from your browser. So what we have done here, we have uploaded a rhodococcus assembly, and with gene calls generated by the PNNL. So what you needed to upload this genome is the a GFF file with the structural annotations and an assembly. So having those two files, you can upload the rhodococcus genome into this narrative, and that's what the first app showing here. And then, so you can see the assembly file here, and then selecting the GFF file here, and then you can upload that two files, and it will be saved as a gene object. And you can, this is where all the objects are. So if you search for rhodococcus genome here, and you can get that genome. So then what we have done here is if you go to the rhodococcus genome that appears in the narrative, so if you go to the browse features tab, you can see all the feature IDs here that got uploaded, but you don't see the functions. So next we annotate the genome with RAST annotation algorithm. And what RAST does here is keep the original gene calls and do the functional annotation of this genome. So it associates functional annotation like pyruvate synthase to those feature IDs. So when a genome get annotated through RAST, And then now if you go to the browse features, now suddenly you're able to see now the function field is propagated for many features, and you can see very useful annotations that has EC numbers. So those are the functions that really relevant for metabolic model construction. Similarly, we annotate with multiple annotation algorithms. So GLM for EC is another annotation algorithm that associate EC numbers to feature IDs. And then I'm going really quick, I apologize to save time. And then it's also annotate with DRAM annotation algorithm. And the DRAM annotation algorithm is a really cool annotation algorithm that associates KEGG orthologs and also EC numbers to annotation. So, and finally, it's annotates in the initial steps, it's annotated with the PDB function. So what the PDB functions that import PDB metadata into KBase genome, that what that app does is it looks for any predicted structural evidence for the features that available in the genome and then associate that data. So what I want to show is that if you go to browse features, now we annotate the same genome with multiple annotation algorithms, and this ontology field should show different annotation ontology events. So let me see that whether I can pull that out really quick. I'm going to search for EC. All right. So what we are seeing here is that on the ontology column, that you can see the annotations that came from the GLM for EC, annotation that came from DRAM annotation algorithms, and the annotation that came from RAST, this associated with SSO subsystem ontology. So multiple annotation algorithms try to annotate the same features. So you have a better chance of getting human readable annotation for each feature. So with that, I will pass to Bill, if you're around, to explain the Snekmer annotation from here on.
Get here. Okay, I'm going to give a couple of slides just on the Snekmer algorithm before we get back to the narrative. How's that looking? Is that looking okay? All right, great, thanks. So I'm going to talk to you a little bit about our sort of evolving functional annotation tool. It's called Snekmer, in which we're using a degenerate K-mer approach to do functional classification of proteins. And so one of the reasons that we felt the need to develop yet another annotation tool is that still we have many bacterial proteins that can't be assigned a specific biochemical function. And so this bar chart is just sort of like a sketch demonstration of what the issue is. So on the left-hand side there you have E. coli, which is probably the most well-characterized organism on the planet. And if you just run it through a simple KEGG annotation and look at both the KEGG ortholog assignments that you get, but also the assignment of those KEGG orthologs to pathways, then you can see that only 75% of the proteins even get a KEGG orthology assignment, and even less than that, about half of that, you actually have some clue as to what the function of the protein is at a cellular level rather than just a particular biochemical level. And this problem gets even worse if you move away from E. coli. I did run another genome. At this point, I can't remember which one I ran, so I just labeled it as other. But that one had significantly less. So somewhere in between those two extremes probably lies the truth for any organism that you in particular are interested in. So we developed Snekmer with two concepts in mind. One is a reduced character set that we use to essentially recode the protein sequence of the proteins that you're interested in, leveraging property similarities between amino acids, and I'll describe that in a little more in just a second. The other part of Snekmer is Kmer profiling, that's the way we actually do the protein comparison, so there's some computational advantages to that. So the reduced character set will simplify your computational problem. And it also can account for sequence diversity, both observed and unobserved sequence diversity in the sequences that you're interested in. And the Kmer profiling, as I said, leverages your computational efficiency. So you get a reduced computational time, you have reduced effort in maintaining the models that you build using Snekmer, and depending on what you're looking at, you can actually increase your search sensitivity, particularly for fragmentary sequence data, and we're still maintaining the specificity of assignment that you get from this tool relative to other tools. And I will note Snekmer has been published. I didn't put the link on here, but I do have it in the presentation that I'll be giving tomorrow afternoon. So the reduced amino acid recoding that I was talking about is laid out here. These are the alphabets that we have available currently. You can, if you have no recoding, then you just have your 20 amino acids that your proteins are usually represented as. But what we're doing is essentially we're grouping amino acids based on similarity of properties. So we have everything from an 11-state alphabet that's based on observed substitution frequencies, the chemical properties, down to a very simple two-state hydrophobicity alphabet categorizing amino acids as either hydrophobic or hydrophilic. So this is what the process looks like if you want to make a model. And this is one of our newer additions to Snekmer, which is the Snekmer Learn Apply. Thank you. Snekmer Learn Apply functionality. So you start with a training set, and this training set is a set of proteins that are labeled by a family that they belong to. So in this case, let's just say labeled by Tiger Fam ontology assignments. Or something like that. So you have your protein sequences, and the protein sequences are recoded according to whichever alphabet you choose. In this case, this is just demonstrating what happens if you recode this sequence into the hydrophobic, hydrophilic character space, and then that sequence is Kmerized, basically broken down into its constituent Kmers. And then we simply make a counts table. Each Kmer, each family, how many times that Kmer is observed for that family. And that's basically the learning part. So this is how we build the database against which you will search your genome. For the actual searching part, we call that apply. And the same thing goes for the proteins that you input. Each unknown protein is going to go through, get translated into the new alphabet, become Kmerized, and we just generate a count for that. And then we compare the count vector that we prepared for the unknown protein against all of the protein families that we previously had processed. And for this, we're using cosine similarity. So this is just a representation in three-dimensional space showing the vector directions of different protein families that we'd already done, and then our unknown protein here. If you can imagine a much more highly dimensional space for all of the Kmers that we might have observed. Oops. That's what it actually looks like mathematically. But whatever vector is closest to your unknown protein, you get a score for all of that. And based on these scores from the cosine similarity, you can classify your protein, your unknown protein, into a specific family. So that's how the Learn Apply is working. And this has been implemented in KBase in a very simple interface, as you might imagine. For input object for this, you can either input a protein sequence object set or a genome object. At the moment, you have to indicate whether it was a protein sequence object or a genome object that you input. We've currently got 3 different libraries that you can search against to do the annotation: TigerFAMS, PFAM, and Panther. And the runs don't really take that long, which is kind of nice. And what you get back if you put in a genome object is a genome object that is now annotated, that includes the ontology terms that Snekmer is applying. And you also have a table format that you can download in a zip format. And so the genome object that you get back will look something like this, where the CDSs is the object type within the genome model that are going to have the annotation. In this ontology column, you will see here, you can see that if you do multiple runs against different libraries, those will just get appended into your genome object. So let's see here. I think I can just go over to the narrative at this point. Okay. That is not showing up on the screen. I'm going to have to unshare and share again. Okay, coming down to annotation trend, Oh, here it is. So we'll just reset this. Like I said, here we have the simple interface with our rhodococcus that we're pulling in here from the list. It is a genome that we're putting in here, and the annotation that we will apply. Let's just for fun switch it up and run Panther on this. We'll see what happens when we do that. And I suppose we don't necessarily want to wait to watch that run. So this is the search that was run previously. And if you go to the browse features, then you can see we've organized this by CDS type. And you can see that Tiger was run previously, and here are all the Tiger annotations that have been put in there. I believe that currently the Tiger annotations have not been mapped to the KBase reaction ontology, so that right now this annotation is not available to assist in your metabolic modeling, but that is in process and should be available shortly. Is that right, Chris? Yeah. Okay. Okay, so it's the Go terms that will be the mapping point between these ontologies that we have for Snekmer and the metabolic modeling. And so that is my demonstration of Snekmer. If you have questions, I can take those later, but now I will turn it over to someone else to talk about importing and merging annotations from external tools.
All right, I am Patrick the Hassler. I am not Patrick the Hassler. I'm Chris Henry. I'm presenting for Patrick because he got hung up in the middle of the country on his way here. So he is not going to be here on time, but he will be here. So if I do a bad job, you can ask him lots of questions later. There are a lot of slides here, so I'm just going to zip through them rather quickly. Basically, the idea is that we've been working, the Lawrence Livermore team has been working with the KBase team to develop a probabilistic annotation pipeline. So the idea is we've got all these different annotation tools to annotate genes. We've got a lot of genes that don't have annotations at all, and then we've got a lot of genes that do have annotations, but the different tools disagree about what those annotations should be. And so that could be helpful because you know, the annotations become theories that can fix gaps in our models. So what the tools in KBase today let you do, for example, is build a model from one particular set of annotations. Currently, that's limited to RAST for the ModelSEED 2 app, but eventually you'll be able to pick other annotations as well. But you build a model from one set of annotations, and then you can gap fill from the other set of annotations. So then all that, you know, all those disagreements become valuable because they give you theories that can be used to fill gaps. And that's what they've added. The Lawrence Livermore team has added these tools to import annotations from staging, so you can import external annotations. So you're not limited. Currently, what Jonica showed you this morning was a bunch of tools that we've loaded in the KBase already to annotate genomes. And if you look through the narrative in detail, you'll see all of those tools. But you can also run tools outside of the pipeline, and that's what I'll demonstrate today, and import those annotations with probabilities, so with evidence. So that's the new thing that is coming in this year is really the ability to map evidence on the annotations, which then influences the weight that the various algorithms have in adding annotations to, and their annotations being used by the modeling to explain phenotypes. And then there's an app to merge annotations together and then use like a Bayes model to merge probabilities from multiple annotation algorithms into like a consolidated algorithm. So then you get this notion of consensus from different algorithms. Like if many algorithms agree about the function of a gene, that function should have more weight. So again, many algorithms disagree. We're looking at using Bayes rule to combine probabilities from many algorithms. This is what Bayes rule looks like. To get these probabilities, that's the first challenge we have, is how do we figure out what the probability of annotation should be. We've chosen to use SwissProt, specifically evidence associated, so like validated, experimentally validated functions in SwissProt. That is our gold standard. So we're evaluating everybody's annotations against SwissProt. And one of the things we're working on, we don't have these tools in KBase quite yet, but these will be coming soon this year, probably in the next couple months, is actually an ensemble modeling pipeline. So what we'll talk about today is more of a integrate it all and make one good model that fits the data as good as possible. This is a different approach where we build many, many models and we test them all and we can evaluate how they perform against one another and then pick some set of winners and reconcile and make a consolidated model that way. This approach can be complex and it can blow up the possibilities, but it can discover solutions that the single optimization approach will miss. So these are the apps that we'll have when the ensemble modeling pipeline is complete. So you'll be able to build an ensemble modeling from these annotation probabilities. So it'll sample the probabilities. Then you gap fill. So then it will reconsolidate ensembles based on the gap fillings because it will pick from the annotations that were rejected by the sampling. And then you run the ensemble, do FBA. So the idea is we'll make better models from this. And this is building on foundational work that Jason Papin started. Lots of challenges. And one of the biggest challenges we've had that I'll dig into a little bit here is just the ontology mapping. So ontology mapping has been a real struggle. Fortunately, some of the tools that Rick talked about this morning are going to really help us with this. So we're looking at using, for example, ProteinBERT, a human natural or not ProteinBERT, BioBERT, to do human natural language modeling so we can consolidate. BioBERT has the ability to look at two different names for a function and say, oh, you're actually referring to the same thing. Those two names are two different set of words for the same idea. And that's working really well. So I think we're about to see a revolution in our ontology mappings in KBase as we are able to complete that work and pipe it into the platform. And then we'll make a nice visualization of the ensembles to see how well they cluster. And then next step is to finish these apps and get these probabilities assigned to as many annotation algorithms as possible, and then see how they perform when we go to fit our data. So with that, I'm just going to quickly show you what the apps in KBase look like. Got the narrative open already and already set to my step. So this is the step 3 of the narrative. To import and merge the annotations. So it starts by exporting your genome. So this is the Genome Object Info app, which lets you export a model as a protein FASTA. So once you have a protein FASTA file, so you just run this app and you get a protein FASTA file that you can just download directly here. So you get a download link, you grab that file, and then you can run all sorts of tools outside of KBase. So we've done Blast Koala, DeepEC, DeepGo. In this case, we're doing NetGo, which is a web service. So we're submitting this to a web service, getting the annotations back. Then you run the import app from state. So you load your annotation file, which ends up looking like this. It's just a tab delimited file with the first column being the gene ID, the second column being the term that was annotated, the Go term, and the third column being the probability. So if you have a tool that gives you a probability, definitely get those in, because the probabilities go into the modeling, the OMEGGA method that Hyun is talking about. So it's going to use those probabilities to give these different annotation sources different weights as it's picking which annotation source it's going to use to fix holes in your model. So you upload this to the KBase staging area. So you just literally just drag your model into the import drag box. And once you've uploaded it, you run the import annotations from staging, and then it tells you, you want to check this, always check this. Please look at the output when you run KBase apps. You guys like build models that don't have any genes in them and then write papers about it. And it's, yeah, check your KBase apps, check your output very carefully. Make sure that your results make sense, because it's easy to misuse the tools. We try to make them as, you know, we give you a lot of rope because we know you want to try new things. And so but that can be a double-edged sword. So just make sure you check your output. And then the nice thing is this compare metabolic annotations app. This actually tells you, because of that ontology mapping, it tells you how well all the different algorithms that you've run, how well they agree with one another. And it's really interesting, the output here. I have 2 minutes. Okay. It shows you the number of distinct gene reaction mappings that come from the method and the number of ModelSEED reactions that were actually unique that come from the method. So when this number is bigger than this number, it means the same ModelSEED reaction is landing on multiple genes. This shows you how many terms are annotated by the method. And the thing I want to point out here is we have a variety of tools, and they all have different attributes that make them valuable for this purpose. So some things will say if it quacks like a duck, it's a duck. And then there's some algorithms that say if it's got feathers, it's a duck. That's okay, because this is the source of theories. So we have algorithms that are extremely aggressive with their annotation and annotate everything with everything. But it is a nice, but they are, you know, typically tend to be right on the third level, the EC classification system. So they are a good source of theories about what the functions of a gene could be. But then you've also got algorithms that are fairly conservative and will leave a lot of stuff unannotated, but will annotate the things they do annotate, they annotate fairly accurately. So this is important for the probabilities because we think about recall and precision. We think about both sides. How many false positives do you get? How many false negatives do you get? And you know, this is not, I am not at all criticizing GLM4EC. I love it because GLM4EC fixes a lot of my gaps, because it does annotate a little more aggressively. So it's those longer distance annotations that are often the source of gaps. So that is it. Oh, there is the final app is the merge metabolic annotation, which makes a new annotation in your object that consolidates the probabilities from all the other annotations. So eventually what you can do is, currently can't do this in the ModelSEED 2 app, but eventually you'll be able to specify exactly what annotations from your genome you want to use both to build the model and to gap fill the model. Currently, it just uses RAS to build the model, and it uses all of the others to gap fill. You'll be able to specify exactly which one, which is where this app comes in, so you can consolidate a bunch of annotations, get a consolidated probability, and then use that consolidated probability to build your model. So with that, I'm going to hand things over to Hyun and Jeremy for the final section.
Right, I'll be talking about the OMEGGA pipeline that we co-developed with the PNNL Soil Microbiome SFA, KBase team, and University of Nebraska-Lincoln. So I'm sure that you are already familiar with how genomes is annotated and to build a model, and where the gap filling process is critical to improve model accuracy. I don't know how. And typically we use, as Jenica initially said, typically we use the parsimonious approach to identify minimal set of reactions to be added to fill the gaps. And then when there are multiple phenotypic cross observations, then we gap fill one at a time in a sequential manner. So if you look at those reactions that are added by the parsimonious approach, they are not necessarily biologically relevant. and consequently, the resulting metabolic models may show some inconsistency with experimental data and phenotypic observations. So OMEGGA app, really OMEGGA algorithm, was proposed and developed to resolve these issues. So OMEGGA carefully examine or evaluate biological relevance of reactions to be added for gap filling based on the multiple genome annotation outputs. Also, it's looking at the omics data, such as transcriptomic data and proteomic data, metabolic data, and so to preferably select reactions that are highly associated with those measurements. And another interesting piece of OMEGGA is the global GApfilling. So instead of GApfill one at a time in a sequential manner, and we seek a set of reactions to be added for GApfilling that simultaneously fit the multiple phenotypic observations. So as the KBase already has apps for model building and GApfilling, so instead of building new apps, then we decided to incorporate OMEGGA algorithms into existing KBase apps. So in the workflow here, then I have highlighted what additional components were added to the existing apps. So on the left, you can see in addition to REST annotation, so we can incorporate a bunch of different complementary annotation pipelines. So at the moment, we focus on those annotation pipelines that are running in KBase, such as TRAM, PROGA PDV, and even Snekmer in the future. So also it will be extended to the other external tools such as Koala and Pathway tools. So there are two ways to integrate omics data. So through at the stage of model building or through GApfilling process. The reason is that because model building app also allows for GApfilling as an option, so that can be chosen as an option. So for GApfilling, now you can see global GApfilling is available as an option, but for integration of omics data, so we can choose options, whether it'll be integrated through sequential GApfilling or global GApfilling. So sequential GApfilling function was already there, but it's not exactly the same as before because that all the process is streamlined through automated process. Specifically that we have two new apps out of this work, and so including MS2 model building with OMEGGA, also MS2 improved GApfilling with OMEGGA, and it has these two apps have similar inputs, but not exactly the same though. So specifically, I highlighted what additional inputs it can take compared to previous versions. So we can incorporate multiple genome annotation through genome or genome set input. Here also you can additionally take expression data as additional input. And then also on the right, you can see that there are three different options for GApfilling, including sequential and global I already talked about. Also, there's independent GApfilling. So I've not talked about it, but because this is independent GApfilling without integration. So we do this for comparison purpose. So we previously demonstrated and tested our algorithm using model organisms such as E. coli, and then in the figure on the left, you can see y-axis is biological relevance of gap-filled reactions, and x-axis different approaches. You can see blue box over there. It shows highest biological relevance. That is the result out of OMEGGA, applying of OMEGGA algorithm that integrates multiple annotation pipeline and transcriptomic data. So we are now extending our analysis to non-model organisms using 8 isolates of model soil consortia for MSC2 developed by PNNL Soil Microbiome SFA, and then it needs phenotypic data from multiple carbon sources as shown here, and green and orange represent growth and no growth. And also we have a transcriptomic data to integrate. Before showing the demonstration using the KBase narrative, I'd like to get your attention to other related presentations here, and from which you can get additional perspectives of OMEGGA algorithms in connection to other related work. So including the poster by Jose on Tuesday is already going on in parallel poster session. So, but there are 2 more oral presentations on Wednesday from Chris and Bill. And then there is another on Thursday through KBase, another KBase workshop, and where Christian is participating in. All right. Now I'll pass it on to Jeremy, Jeremy cannot attend in person, but he's connected through Zoom, and then he'll go over OMEGGA KBase narrative, then to demonstrate OMEGGA. And then Amy Castle on the right is my students who also made a significant contribution to the development of the KBase narrative. So at the end, they will have us Janica, and then to share how ActionMap can help us to see the gap filter reactions on the pathway level.
So I'm going to be continuing just where Chris left off in the demo and going through the steps that Dr. Song spoke about. And so really the first step is just adding in a couple more data types. So previously we had the fully annotated genome, and now we're going to add in media type and phenotypic growth data. And so if we just open this right here, we can just see ones and zeros based on if Rhodococcus grows under different media types, and it grows under 9 of these media types. And so Rhodococcus is our demo data that we have. And from this, we are going to build our draft model. And for this model, there's an option to gap fill it immediately, but we're going to save that to the next step. And what this draft model essentially is it is a fully annotated genome that is essentially restructured to display data a little bit more easily and to allow it to be gap filled. And so right now there's no gap filling that's occurred. And it just takes that input from the previous step. And with this draft model, we are going to first check just how it performs prior to any gap filling. And by simulating growth using a flux balance analysis, we could see that it essentially it doesn't predict it to grow under any conditions, which I mean, we know phenotypically and through test data that it does grow. However, based on our annotations, it says it doesn't grow. And so that's an indication that we have to do some gap filling because there is some missing data there. And so first, I'm going to show 2 options here. So we've got our sequential gap filling. And this is running across 9 carbon sources, which are those ones that I showed above. And after running across these, we can see that it adds 70 reactions, and it is 75% accurate overall. And these results can be seen using the flux balance analysis after we've run the gap filling. And we've got a minute, so I'll just show. This is what the configuration for the gap filling looks like. So you just select your media types, you gap fill, and it runs all of them sequentially. And then the other one that I'm going to show you is very similar. It's sequential gap filling, but also has omics integration. So this has transcriptomics data from Rhodococcus for 3 replicates of glucose, 3 replicates of NAG, and then 2 of R2A media. And so with these conditions, we got similar results. However, when you consider that there's only really 3 media types here versus 9 above, and we're getting pretty similar results, that's an indication that the addition of transcriptomics data is helping this find more reactions. And results again can be seen in the flux balance analysis. And I'm going to pass it on to Jonica, who's going to speak about the Escher pathway map for the remaining time.
And then click gap filling. Click it twice. So what you see is you have gap filled reactions here. That's what That's what it means in gap filling, and it lists why the reaction was gap filled. It lists the condition that led to the reaction being added. The key thing is because we have those probabilities and those other annotations, you can see we now have, for a lot of our annotations, a gene candidate. So that's, in some cases, for many of the reactions, like most of the reactions. So we now have a gene candidate. That's what it's doing when it's integrating those annotations. So if you have a really aggressive annotation algorithm with lots of false positives, it'd be nice if you had a way to cherry pick the false positives and find the ones that fit with your other experimental evidence. That's exactly what this app does. It's like, oh, I don't like most of these, but that one actually explains growth on this phenotype. So I'm going to use that tool. When you add the expression data, it helps it to do a better job of that because it's now using more experimental evidence to pick the right gene to fill those gaps.
All right, so the last part of this presentation is probably the fun portion of this presentation. And now you listen to how the gaps being filled in the metabolic network. Now, how do we visualize these gaps? Sometimes you have this network, you're really interested to know which pathways were gap filled. Is this organism lacking histidine biosynthesis, for an instance? And you suddenly want to see that which pathways are lacking the initial reactions and which pathways were gap filled. So this app called the Asia Pathway Viewer, what we are doing here is now we input the metabolic model that was gap filled, and we also have the flux analysis data into this app, and what it produces is an Asia map. And this Asia map, so the app output looks like this. I can open this Asia map in a separate window, which shows me a larger version of this app. So this will show also the fluxes of this organism when it was simulated, and also with very bright green colors, it also shows the gap filling reactions. And this is just for the heme and seroheme biosynthesis. And I have a link down at the bottom of the narrative that will also show a genome scale version of the Asia map, which is more interesting. Let me see, I have that. I'll just open this one here. All right, here we go. So that you can see this, there's lots of reactions, and I can open this in a separate window as well. And that will show in the genome scale which gaps were filled with the bright green color in different sections of the metabolism. And so this was a close-up, and so in one pathway that will show the what reactions were gap filled. So with that, we conclude the presentation, and if anyone has questions, just feel free to reach us, and we are happy to answer any questions.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.